Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maraj, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Cybono Discovery Center. Today, we are going to be discussing the paper 1 revision for the paper that you will be writing tomorrow. Yes, it's already that time when it's time for Life Sciences paper 1. For the preparatory examination. And I am together with my colleague and friend, Tiniko Vuma, who will be also presenting this lesson with me. So I just want to start with saying that the preparatory examination is very, very important. I'm sure you have seen all the papers that you have done so far are there and designed for you to be prepared for the final examination. So it's very important that you take it seriously and that you really put a lot of effort into studying for it and make sure that you know what is happening. Now, I just wanted to explain to you that it is not possible for us to do all the content and to reteach everything that you have learned for paper 1 in today's revision lesson. We will be focusing on some important aspects and in some concepts that are normally confusing for learners or topics that learners usually struggle with. We'll be focusing on those things. And we have also designed our presentation from the topic with the most amount of marks to the topic with the least amount of marks. And I want to also explain to you grade 12s that this does not in any way make the last topic any less important than the first topic. It's just the weighting of the amount of marks. So obviously there is more to study and there's more content around the topics that have a higher weighting or a higher mark allocation. So this is our paper 1 breakdown. We have our reproduction in vertebrates at 8 marks, human reproduction at 41 marks, responding to the environment in 54 marks for humans, responding to the environment in plants, 13 marks, and endocrine and homeostasis at 34 marks. So today we'll be starting with responding to the environment, and then we'll do human reproduction, endocrine, then plants, and then ending off with our reproduction in vertebrates. Right, so let's get right into it. The resources that you need. So these are 2 small QR codes for 2 small little links, but Dynamite comes in small packages. There is a wealth of information and resources that you will find in the Learner Resources Google Drive folder, and as well as our Life Science Gauteng Instagram page. So please make sure that you are going onto those platforms so that you can see past papers, TV lessons, lots of resources in this one. And on our Instagram, this is where we share valuable information, little videos, little tips for the exams, and things like that, right? So human response to the environment yes. What does the examination guidelines say? So these are the topics. I'm just going to bring them all up over here. These are the topics that you are needing to study, the main things. This is just the main things. The brain, you need to know the structure and functions of the different parts of the brain. You need to know about the spinal cord, the structure of the spinal cord. You must be able to describe the reflex action using a diagram. Then you need to know the neurons. You need to know what neurons are. You need to know the 3 different types of neurons, as well as their functions. And you also need to know how to label them. You need to know how to draw them. Very important. And then we have the human eye, which is an organ, right, of the nervous system. You need to know the structure and the functions of the different parts of the eye. And then you need to know about accommodation and pupillary mechanism, right? And then we need to know the visual defects and the treatments of them. Then lastly, we have the human ear, where you need to know the structure and the function of the different parts, the process of hearing, what happens during balance, and lastly, the hearing defects and also the treatments of them. Okay, so that's a little overview, a quick overview of what the examination guideline says you need to know. Okay, and yes, reminding you that this is 54 marks. Okay. Hopefully, you've already started studying for life sciences, but if you didn't, then this is definitely the topic where you should start. Okay. So here we have two different parts of the nervous system. We've got the central nervous system, which is made up of the brain and the spinal cord. And then we have the peripheral nervous system, just reminding you that it's made up of the motor pathways that we are going to look at, which are the somatic and the autonomic nervous systems. And our autonomic nervous system is divided into our sympathetic and our parasympathetic. Please don't forget about this. I'm not going to do it today, but I need you to still remember. And please, like I said, we are not doing all the content, so don't think that this is the only things that you need to study. You need to study every single bullet that is in your examination guideline. Okay, so here we have the cerebrum and its function, the corpus callosum, the pituitary gland, the medulla oblongata, the cerebellum, and the hypothalamus. So like I said, you must be able to know how to label this diagram. You must be able to know what are the functions of the different parts, and you also need to know where the different parts are located. You need to understand where the cerebrum is. You must be able to describe where the cerebrum is, where the hypothalamus is, where the corpus callosum is. You must be able to be well orientated with where the location of these different parts are. Also, examiners can give you a scenario, right? For example, they might not blatantly ask you, what is the function of the cerebrum? Or they won't just ask you, name two functions of the cerebrum. No, they could make it a little bit more complicated. They can maybe say, Susie's reading a book. Right? So when Susie's reading a book, right, then you must understand that that is a higher cognitive functioning, and it is going to be controlled by the cerebrum. Okay, so they can ask you which part controls the fact that she is comprehending what she is reading, or which part of the brain is going to control Susie lifting her arm. Okay, that's a voluntary action. It's also the cerebrum. Or they can ask you which is the center that they can tell you that they can give you a scenario about somebody kicking a ball or doing something like that, and then they basically give you information or imply that it's about balance. Then you must know that balance and is the cerebellum. Okay, so please don't confuse the cerebrum with the cerebellum. Learners often confuse these two. They are different structures, completely different structures with different functions. Then we've got the three types of neurons that I mentioned to you: the motor neuron, the sensory neuron, and the interneuron. Please take note, we do not use the word really connector neuron anymore. So please use interneuron. You need to know how to draw these neurons. I'm going to write here draw so that you can be reminded. Please make a note. You need to know how to draw them. You need to know how to label them. And please, you need to know how to label them and the structure, and the functions of these particular parts that are mentioned here. Look at your examination guideline. The structure that they mentioned there of the neurons is what you need to study. The textbooks have many other labels and many other structures that are labeled for the neurons, but you only need to know the ones that are stipulated in the examination guideline. Okay, take note of where the cell body is. Take note of where the dendrites are. Okay, understand what is happening in these three diagrams. Okay, one more thing I wanted to say about this, which leads us to our next part. You also need to understand what a receptor is. Stimulus first, sorry, what a stimulus is, what a receptor is, what is the functions of them, okay? And then you also need to know that it is an impulse that is transmitted. An impulse is transmitted. It's not a message which is sent. It's not a message that is carried. It is an impulse, okay? So the stimulus is the one that is the change in the environment, okay? This part I have to mention, otherwise you don't understand anything that happens in the nervous system if you don't understand that. So the stimulus is a change in the external or the internal environment. The receptor is the one that detects the change, right? And then it is going to convert the stimulus into an impulse. Yes, what impulse is that? A nerve impulse. And then the neuron is going to transmit the nerve impulse. Okay. So the sensory neuron transmits the nerve impulse to the central nervous system, and then the motor neuron is away from the central nervous system. Which two parts make up the central nervous system again? The brain and the spinal cord. Very important. Right, now we move on to reflex action and the reflex arc. The reflex action is a quick automatic response to a stimulus. So have you ever touched a hot plate? Okay, you will be reminded of that. We did this in class already. I'm just reminding you, it's the one where you have a quick reflex reaction. It's the one that is going to prevent further harm. So it is there. The point of it is there so that it prevents further harm. So for example, you're going to touch a hot plate. Okay, your hand is going to automatically move away in very, very quick response to prevent yourself from burning further. So the examples of these are the knee-jerk reflex, sneezing, quickly moving a body part away from harm. And then what is the reflex arc? It's the pathway along which an impulse is transmitted to bring about a response to a stimulus. Grade 12, it's very important that you know how to distinguish between a reflex action and a reflex arc. So I always like to explain it in this way. The reflex action is what is happening. What is it? It is a quick automatic response to a stimulus. That's what it does. So reflex arc, what is that? It is the pathway. It's the pathway along which the impulse is transmitted. Reflex arc is the actual arc, the pathway, and the reflex action is the response. What is the significance of it? Like I said, it allows for a quick response to prevent damage to the body and also to prevent further damage. Yes, you are going to feel that pain. Sometimes you do feel the initial pain. You will be pricked, so you will have harm, but it's also to prevent further harm. Because obviously, if you're going to keep your finger or you're going to keep your hand on that hot plate, you're going to burn very badly. Right, and then just to remind you, we always talk about an impulse and never a message. Grade 12s, you need to know this pathway. I'm putting an NB here. You need to know this pathway. You need to know the stimulus. The stimulus here is the hot object. You need to know the receptor. The receptor is now in the finger. Okay, you need to know that it's going to be taken via the sense. The receptor is going to convert the nerve impulse. Convert the stimulus into an impulse. Travel via the sensory nerve, and then you have to know that it's going to enter the dorsal root of the spinal cord. It's going to make a synapse with the interneuron in the gray matter of the spinal cord, which is then going to have a synapse with the motor neuron. The impulse is then going to travel via the motor neuron to the muscles. The muscles are going to contract, which is going to bring about the response. What is the response? Moving the arm away from the hot object. Now, they can ask you anything. They don't have to ask you about the hot pot. They can ask you about any other one, okay? So you must be able to, if they give you a diagram and they ask you to explain what happens, you need to see what is happening in the diagram, because some reflex actions do not have an interneuron. They don't via the interneuron. Okay, there isn't one in some of them. So please always be very vigilant and analyze diagrams that are given to you, analyze information that is given to you before you answer the questions. Okay, then we are moving on to the internal structure of the eye. I'm not also going to go through all of them, but this is what you need to know. You need to know all of these ones. Okay, so please make sure that you understand all of these. You should already be having diagrams in your bedroom, pasted on your walls that you are supposed to be looking at every single day. Okay, child, if you don't have these kind of structures, right, now is the time when you are starting to start focusing on making them. in preparation for the final examination, where you have a picture, even if you don't make it yourself, you print this on a big A4 page or an A3 page, and you have it stuck up in your room so that every time when you wake up in the middle of the night, you're looking at the eye and you are trying to recite the different parts. Okay, so please make sure that you know all of these: the ciliary body, the suspensory ligaments, pupil, iris, lens, blind spot, optic nerve, yellow spot. You need to know how to label this diagram. You need to know the functions of all the different parts. Structure and function, very important. And I don't just mean, when I say structure, I don't just mean you must be able to label them and know the part. You need to know what are the properties of those parts. So the lens, okay, what are the properties of the lens, the characteristics of the lens? It's elastic, it's transparent, it is biconcave. So that is describing the structure. You need to know that. Examiners also like to ask you to compare. They like to ask you to compare, for example, the structure of the lens with the structure of the sclera. How are they different? Okay, so you must be able to know your work. You need to know all the structures. You need to know what they look like, what they feel like, what is their shape, and what their functions are. And then that brings us to the pupillary mechanism. Okay, what is the pupillary mechanism? It's a reflex. Yes, it's a reflex, and it's in response to the stimulus, which is light. So if the stimulus is either bright light or it is dim light. Yes, you should know this already. Okay, so I'm reminding you that in the iris, I'm first reminding you of the parts that's involved in the pupillary mechanism. They are the iris and the pupil. And within the iris, we have circular muscles and radial muscles. Don't get confused with ciliary. We are talking about circular. So we have circular muscles and we've got radial muscles. And those two work antagonistically. Okay, so they work in opposite directions to each other, and they control the size of the pupil. So when we have bright light, it's very, very bright. Like in the moment in the studio, I've got bright lights focusing on me, right? So our pupil will become smaller, right? You should know this. Why? Because the bright light can damage the retina. So it needs to become smaller to allow less light to enter. We don't need a lot of light to be able to see. Right? So in bright light, the pupil is going to constrict, and in dim light, it's darker. So you're going to need more light to enter the eye. So the pupil is going to dilate. Very important that we use those words: constrict and dilate. Constrict or dilate. Don't use other words because they mean other things, right? So to remind you, in bright light, the circular muscles of the iris contract. Of the iris, grade 12 is very important because these muscles, we need to know that you know that these muscles are in the iris and that you know that we are talking about the iris here. So the circular muscles of the iris contract, the radial muscles relax, and the pupil then constricts. The amount of light entering the eye is reduced, or we can say less light enters the eye. And this is where we get our little mnemonic over here. Circular muscles contract, radial relax, pupil constricts, less light enters the eye. Okay, so study the one, because the dim light is going to be the other one. If you find dim light easier for you to remember, you must study dim light off by heart, and then you must know that the bright light is the opposite. Okay, so in dim light, the circular muscles of the iris relax, the radial muscles contract, the pupil dilates, the amount of light entering the eye increased, more light enters the eye. Then we've got accommodation. Okay, accommodation is about seeing near or far. Okay, so you're either seeing an object less than 6 meters away, which is near vision, or you're seeing an object more than 6 meters away, which is far vision, right? Remember, you've done this already. I'm reminding you now. What are the parts that are involved with accommodation? It's the ciliary body with the ciliary muscle. It's a suspensory ligament, and it is the retina and the lens. Okay, those are the parts that are involved when it comes to near and far vision. Okay, so you need to know it. With far vision, you need to know that the ciliary muscle is going to relax. You can see it's put out nice over here with some red typing as a text as well, so that it can help you to remember. The ciliary muscle relaxes. See, ciliary muscle, not circular. That's where learners get confused. So in the previous one, I want you to remember about the circular muscle, and I want you to remember that it's of the iris. When we are here, we are talking about the ciliary muscle, which is going to relax. Then the suspensory ligament is going to become taut. So the ciliary muscle, right, it's going to relax. And then the suspensory ligaments are going to become taut, right? Or you can say they become tight. And you cannot say they contract. They do not contract because they are ligaments. Ligaments do not contract, okay? So that's the thing I wanted to mention for you there. You must say that they become taut or they become tight because they are ligaments. And then you're going to cause the lens to become less convex, okay? So I want to just show you the shape quickly. This is biconvex. The shape of the lens is biconvex. It can either become less convex or more convex. It's already in a convex shape. So it's important that we use the correct words. The lens becomes less convex. Now, when it comes to the far one, you need to know that the ciliary muscles contract. You must talk about the suspensory ligaments that slacken or loosen, and then it becomes more convex. I know in the diagram it looks like it becomes rounded, or it looks like it becomes round, or it looks like it becomes fat, or it looks like it becomes flat, or whatever the case means. Don't use those words. Learn less convex and more convex. Then you know that you are going to be 100% correct. Now, as you know, that's how the eye is supposed to work, right? So that we can get a clear image focused on the retina. However, it doesn't always happen, as you know, okay? So sometimes we have an issue with the eye. We have an eye defect. We have short-sightedness and long-sightedness. We're going to go through them quickly to remind you. Short-sightedness is the problem seeing objects far away. Now, I also want to mention to you, I'm also going to put an NB here. And the reason why, I don't know if it's going to come now or the end of the year, but I do know that they've been asking these questions recently about the eye defects and the ear defects, and learners are getting it wrong. Okay, so that's the reason why I chose to explain it today. So it's a problem seeing objects that are far away, right? The eyeball is either too long or the cornea is too curved for the length of the eyeball, or the inability of the lens to become more convex. So there's three issues, sorry. The eyeball is either too long. So if it is too long, or if the cornea is too curved, it's going to affect the diffraction. Okay, so if the eyeball is too long or the cornea is too curved to affect the diffraction, and the lens cannot become more convex, that is going to affect the diffraction. And when the light is going to bend, it's going to cause the image to fall in front of the retina. So that's going to be a blurry image, right? Why, grade 12s? You know this. Because in order for there to be a clear image, the light must fall on the retina, right? So then that's what happens. Now, what is the treatment for this? We have to use a concave lens. Okay, so you need to see, can you see the concave? You can see this shape is clearly concave. It's the opposite to convex. Okay, it's like going inwards on either side. So that's a concave lens, and that causes the lights to be refracted outwards, right? It causes the light, so it's going towards the lens, it's going this way. And when it does that, it then causes the focus to become closer, so that for the focus to go further, so that it reaches the retina, because at the moment it is falling short. Okay. Long-sightedness, on the other hand, is people that have problems seeing close objects. Okay. And this is because the distance between the lens and the retina is too small, or the eyeball is too short. So the light is focused behind the retina. So it's going far back behind the retina. And it's also harder for people to read as they age, okay, because of this reason. Lens loses elasticity and they cannot become convex enough. Okay, age does these things to our body, right? So how do we fix this? As you can see, the lens is falling behind the retina. How am I going to fix this now? So what you're going to do is we're going to use a corrected by biconvex lens. So we already have a biconvex. We're adding another layer of this biconvex shape to cause the diffraction to happen. And so that the converging, it will be now converged. Light is going to move inward and then it's going to be focused closer. Because remember, it's gone behind. Now it's going to be moved closer. Okay, so let's do a quick question. Remember, whenever you see a diagram, you must first label it. I'm not going to do it for you. You're going to do it yourself, okay? And then we need to identify part D. So part D, it's over here. It is the pupil. Part E is the cornea, right? And then we need to give the letter of the choroid. Very important, grade 12s. If they ask you for the letter, they want the letter. If they ask you for the name, they want the name. Please follow the instruction of the question. The letter of the choroid. So the choroid is the middle layer. Sclera is the outer layer. Retina the inner layer. The choroid is the middle layer. So it will be F over here. And then the iris, it will be the color part of the eye, which is this part over here. So it will be C. Name the two different receptors found in part A. Oh, that's lovely. So what is part A? You first have to ask yourself. It is the retina. What are the two receptors found there? The rods and the cones, right? You need to know there are photoreceptors. There are two of them. There are the rods and the cones. They are found in the retina. Give only the letters of three parts that are involved in accommodation of the eye. You see, like I told you, you need to know what are the parts involved in the accommodation. They want the letters, not the parts. So you need to go ask yourself, what did Mrs. Maharaj say? She said it's the ciliary body. She said it's the suspensory ligaments. And what else did she speak about? Oh, yes, she spoke about the lens. Now, you're not going to write them. You're going to go to the letters. Yes, so we have B, and we have G, and we have H. Let's see if we are right. Yeah, B, H, and G. There we go. Right, then we have the last one for the nervous system, the ear. I don't have it here, but it doesn't mean you mustn't know it. Grade 12s, you need to know the structure and the function of all the different parts of the ear. You also need to know the different parts of the ear if they don't give you the diagram of the whole ear. What do I mean by that? They can either give you the outer ear or the middle ear or the inner ear, but you need to be able to identify what am I looking at here. The stem of the question will give you an indication to say the following diagram represents a part of the ear, so that at least you know which organ we are looking at, right? But they're not going to tell you it's the inner ear or the middle ear or the outer ear, so you need to make sure that you know how to identify them. Then this one is very important, the pathway of sound. Why? And I want to explain to you what the important parts are. So yes, you need to know the structure. Most learners get that right. They know it goes from the pinna, this part. This part, learners know. Pinna, auditory canal, tympanic membrane, ossicles, oval window, cochlea, organ of Corti, auditory nerve. But what they don't know is that they don't know which stimulus is found where. So this one is the stimulus. Okay, that is the stimulus. So it's first a sound wave. Okay, then we have a receptor. What does the receptor do? So all of these now are stimulus, because what does the receptor do? We only get to the receptor here, which is the organ of Corti. What does the receptor do? It converts the stimulus into a nerve impulse. Yes, so this is where we have the nerve impulse. And then we have a nerve that can carry the nerve impulse, right? So before that, we have a stimulus. But the stimulus isn't always a sound wave, because it's a sound wave when it's on the outside, right? It hits the pinna, and then it goes to the pinna, travels via the auditory canal, and hits the tympanic membrane. So when you hit the tympanic membrane, it vibrates. Okay, so that's very important. So you must know that from a sound wave, it hits the tympanic membrane and becomes a vibration. Then the ossicles, it causes the oval window to vibrate, which causes the ossicles to vibrate. Oval window, and then we have the tympanic membrane causes the ossicles to vibrate, which causes the oval window to vibrate. Now we enter the cochlear fluid. And once you enter fluid, it cannot be a vibration anymore. Okay, we don't say water vibrates. Do we say that? No, we now create a pressure wave. So now we've got a pressure wave in the cochlea, in the fluid, and that is the organ of Corti is then going to convert the stimulus into a nerve impulse, which is going to travel via the auditory nerve to the cerebrum, so that sound can be interpreted. Very important. There are two ways to regulate balance. Grade 12 also very, very important. Okay, there are two ways that we have for balance. Always you need to know what the stimulus is. So there are two different stimuli here. It's either a change in speed and direction of the head movement, or we have the position of the head in relation to gravity. Okay. So speed and direction of head movements is like, for example, if you are standing and then you are running, or you start running and then you change the direction of where you are the way you are running. Okay, that is speed and direction of the head. But position of the head in relation to gravity is, for example, if you are bending forward or you are bending back or you are moving your head in a direction, in a certain direction, that is about the position of the head in relation to gravity. Do you understand that? So if I'm bending down to pick up a box, that is position of head in relation to gravity. So if it's about the change in the speed and direction, that's the first thing that you need to identify. The examiner isn't going to always tell you that this is what happens. They're going to give you a scenario. They're going to say somebody was running, somebody was playing soccer, somebody was bending down to reach a box. You must know what is happening. You must be able to figure out, okay? Apply. So if it's a change in speed and direction of head movement, then the receptor is the crista. Where are the crista located? At the ampulla in the semicircular canals. If it's position of the head in relation to gravity, then the macula is the receptor, and they are found in the sacculus and the utriculus. Very important. So here you will remember SCA for speed, crista, ampulla. Here you will remember PMSU for position, macula, sacculus, utriculus. But both of these pathways, no matter what the stimulus is, both of them, till this point, they are going to differ. But both of them, from that point of the location of the receptors, what the receptor is going to do and the response that it's going to bring about is going to be the same. So both receptors are going to convert the stimulus to an impulse and send it to the auditory nerve in the cerebellum. Why not the cerebrum? The cerebrum is the pathway for sound. That is, the cerebrum interprets sound, but the cerebellum is going to interpret balance. Okay, it's going to coordinate balance. So it sends an impulse to the skeletal muscles to maintain balance, to either restore balance or to maintain balance. Very important is, what did the scenario tell you? If we are restoring balance, so in other words, they tripped and you need to restore balance, then you can say you restored balance. If you are running and you change direction, you want to maintain balance. Okay, so it's very important that you read the information that is given to you about the scenario so that you can decide or you can apply what is actually happening. Very difficult. Lots of learners get it wrong. Okay. Right. Quickly, ear defects. You need to know middle ear infection. Normally antibiotics are used, but in children sometimes it's not effective, and then they have they put tiny buttons with fine holes in them, which are called grommets. Okay, and they allow the drainage of the middle ear until the eustachian tube recovers. Right, so that's usually grommets are sometimes inserted if antibiotics are not effective. Then you also have hearing loss because of the following things. Grade 12, I need you to know this. Hardened earwax, ruptured tympanic membrane, damaged ossicles, damaged cochlea, damage to the auditory nerve, brain damage. All of these things can cause hearing loss. What are the treatments? Either hearing aids or cochlear implants. This one is a hearing aid. This one is a cochlear implant. Right, now we have a quick question. This one is about hearing loss. There are two main types of hearing loss. Type A hearing loss occurs when there is a blockage in the transmission of sound waves and vibrations through the outer and middle ear. Type B hearing loss is caused by damage to the hearing receptors of the inner ear or to the nerves that carry sound impulses to the brain. Grade 12, you must read. always read. Don't just go to this and think that this information is not giving you any information. You read with understanding. Then you go to the diagram. It's telling you here it represents the human ear. Label the diagram. Read the passage, label the diagram. Write down the letter of 2 parts each that may be damaged when a person suffers from. Now, where do learners go wrong here? I can already tell you. They give us the name of the part, not the letter. They don't do 2 parts each. Damage when a person suffers from. Type A hearing loss. Now, if you didn't read the passage, you're not going to know what's type A hearing loss. Go to the passage. Type A hearing loss occurs when there is a blockage in the transmission of sound waves and vibrations. Okay? So which are the 2 parts that are going to be affected? It's going to be part P and part T. Why? Because it's a blockage in the transmission of the sound wave and the vibration through the outer and the middle. So this is the outer, this is the middle, right? For type B hearing loss, it's caused by damage to the hearing receptors. Where are the hearing receptors? In the inner ear. And where more specifically? The cochlea. So then we have to see the 2 parts there are the S definitely and R. Yes, it's the cochlea and the auditory nerve. Okay, so that's how you work it out. Identify part S. S is the cochlea, which we just mentioned now. Okay, describe the role of the hearing receptors when they are stimulated by pressure waves. Okay, so now we have the hearing, they're talking about the hearing receptor. So here we must know it's in the cochlea, and we know that the cerebrum is going to be involved, but they want to know what is the role of the hearing receptor. So the pressure wave is going to come. Remember I told you in the cochlea, it's going to convert the stimulus or the pressure wave to an impulse and then transfer it to the auditory nerve. Explain why a person will suffer from hearing loss if the bones at P are fused. So there are 3 bones at P. They are called ossicles. Okay, and what happens if they are fused? Now remember that they are supposed to vibrate. Each one vibrates and takes the vibration, which causes the next one to vibrate. So if they are fused, they won't be able to vibrate freely, or there'll be less vibrations carried to the oval window. Therefore, we're going to have decreased amplification of sound. Fewer or no pressure waves will reach the cochlea. Receptors in the cochlea will not be stimulated, and there will be no impulse or less impulse will reach the cerebrum. Okay, so basically the ossicles being able to vibrate freely is very important for amplification of sound. And if that does not happen because they are fused, that's going to be limited, and that's what causes the issue. Okay, so that is all that I have for the human response to the environment. Now we're moving over to human reproduction. Right. This slide is also giving you an overview of the examination guideline and all the different parts. And I'm going to go through them quickly to show you what you need to study, but also remind you that I cannot do all of this today. So I'm going to focus on main things. Female reproductive system, male reproductive system, puberty, menstrual cycle, hormonal control of the menstrual cycle, and the development of the fetus. Please make sure that you study all these different parts of human reproduction. 41 marks. Right, male reproductive system, grade 12, you need to know both diagrams. You need to know how to label both diagrams. What do I mean by that? These are both male reproductive system. They are just different views. We have a front view and we have a side view. Okay, you need to know how to label the front view and you need to know how to label the side view. I know they look different, but that's why I have this diagram over here, so you can clearly see the different two. There's just a typo over there. I'm sorry about that. It is ureter. Please make a note that when you are studying this, that you're not going to say ureter. Okay, it's ureter. And then yes, we have all of these different parts, and then we've got the accessory glands. Very important. You need to know the accessory glands. And oh, sorry, we're going to come to that. I just want to show you need to know the front view and the side view and all the different parts, right? Then we've got this other diagram here of the male reproductive system. This gives you a more detailed overview of the different functions. Please make sure that you know all of them. There are terms that sound different, that sound the same, but they have different meanings. For example, epididymis and epidermis. Okay, the epididymis is this structure over here. That's where the sperm cells are stored and they mature. And then we have the epidermis, which is the skin. And then we have urethra and ureter, which sounds similar. Urethra is the tube that goes from the bladder to the outside, and the ureter goes from the kidney to the bladder. So please don't confuse these two. Then the orange block is talking to us about our accessory glands. There are 3 accessory glands: seminal vesicle, Cowper's gland, prostate gland. Collectively, they are called accessory glands, and collectively they carry out a function. Their function is essential for the maturation and motility of sperm. They contain nutrients, they are alkaline, they contain mucus. Okay, together with the sperms, they constitute the semen. So that's the part I want you to know. They don't all have their own function. You need to group them together as accessory glands which carry out this function. I'm reminding you about that. Female reproductive system, also you need to know the front view and the side view. Here's the front view, here's the side view. You need to know them. You need to know the different parts. You need to know how to label it. Structure and function, very important. Then we have, here's the female reproductive system. In this slide, what I want you to focus on is this exam tip over here. Learners must be aware that examiners may ask questions based on a part of the system. So they can just cut out this part and give it to you and ask you a question. Right? They don't have to give you this whole thing in order to label it. That's what you need to understand. And then learners must be familiar with the front and side view of the male-female reproductive system, which I already mentioned. And lastly, learners confuse the endometrium and the muscle wall. So I quickly want to show you. Grade 12, this is the uterus, right? This part over here is the uterine wall. That is the muscle. This is the uterine wall. It's made up of myometrium, right? It's a muscle layer. It contracts. This here is the endometrium. This is the uterine lining. We call it the endometrium. It is the endometrium. The uterine wall is always there. The endometrium is the one that gets built during the menstrual cycle, part of the menstrual cycle. The endometrium is the one that gets built up, and it also is the one that sheds during menstruation. And it's also the part where the embryo is, where the embryo is going to be implanted, and it's going to start developing. Right. So very important for you to know that the difference between the endometrium and the wall. Right, then quickly going into the menstrual cycle, just to remind you, it consists of the ovarian and the uterine cycle, and it progresses over a period of 28 days. You need to know this. You need to know the different hormones. We've got FSH, we've got estrogen, LH, and progesterone. I'm putting this here just to remind you, you need to know the glands that are involved, which are the pituitary gland and the ovary, and as well as then the placenta, which is actually not a gland, it's a temporary organ. And then we have the hormones that are secreted by each part, and then we have the functions of the hormones. Please, please, please know this. Where do learners get confused? They don't know specifically that in the ovary, we have the developing follicle and the Graafian follicle. The Graafian and the developing follicle, which secretes estrogen, and the corpus luteum also in the ovary is the one that secretes progesterone. Where else do learners go wrong? The estrogen is responsible for thickening the endometrium, and the progesterone is in charge of maintaining the thickness of the further thickening and maintaining of the endometrium. And then during pregnancy, the progesterone then is there to maintain the thickness. Okay, but the primary function is to further thicken and to maintain thickening. Right, the menstrual cycle, this graph for the menstrual cycle is the best way to explain it and the best way to study from. Please make sure you know what's happening in the ovarian cycle. You know the hormones that are involved and what happens in the uterine cycle. So these hormones are controlling what happens in the ovarian and the uterine cycle. Everything is controlled by the hormones. This is what controls it. Okay. So that's why I'm saying you need to know the hormone. You need to know where the hormone comes from. You need to know what the hormone does. Okay. And then you can apply it to this, because all of it works because of the other one, and they're all working simultaneously. The ovarian cycle is doing its job at the same time the uterine, excuse me, the uterine cycle is doing its job. Okay, so please make sure you know this. You know what happens between day 0 and day 7, day 7 and day 14. You need to know these main events, day 14 and 28. Okay, so what's the main event that's happening? Just to go through it quickly, is that from day 0 to 7, we have the follicle that's growing, and we have the follicle is still a bit small, and we have the endometrium, which is shedding, menstruation. And then we get the follicle that starts developing because of FSH, and the developing follicle, as it develops and it gets bigger and it develops into a Graafian follicle, it's going to secrete estrogen. The bigger the follicle, the more estrogen is secreted, and that is going to build the endometrium. Then at day 14, so what's going to happen is, just before day 14, estrogen is going to spike, because obviously this Graafian follicle is now big and fat and developed. And inside that we have Graafian follicle, we have an ovum. I've got to mention that, very important. Many learners forget that. That in this follicle that's developing, there's an ovum inside. Okay, then that Graafian follicle is going to now secrete a lot of estrogen. When that estrogen peaks, because it's very, very high levels, it's going to stimulate LH to be secreted by the pituitary gland. And that LH is going to cause the onset of ovulation. So what is that? It's when the mature Graafian follicle ruptures, and then the ovum is then released. Okay, that's at day 14. Where does this ovum go? Here's the ovum at ovulation. Where does it go? It goes into the fallopian tube, and that is the site of fertilization. It's going to wait for fertilization to occur. Okay, then we've got the estrogen. I told you it was building the endometrium. Yes, then we have ovulation, and then that ruptured follicle, LH, has a second function. The first function is to cause ovulation. The second function is to convert the empty Graafian follicle into a corpus luteum. That corpus luteum is a large structure which is going to secrete progesterone. You can see it over there, you see. That's why I said the graph is awesome. Because even if you forget what hormone is where, which hormone is secreted where, the graph is going to tell you. The corpus luteum is going to secrete the progesterone. What's the progesterone going to do? It's going to continue the thickening. It's going to make it more glandular and more vascular of the endometrium. Then, once we are reaching around day 21, remember that ovum was in the fallopian tube. Now it's realizing, okay, the body realizes fertilization did not take place. That ovum did not become fertilized. So there is now no need for the endometrium, and there is no need for the corpus luteum. So what happens is the corpus luteum becomes smaller and smaller, it degenerates, and it stops secreting progesterone. So progesterone levels drop. And when those progesterone and estrogen levels drop, that's what stimulates the endometrium to shed. And then we have menstruation. Okay, so very important for you to know. Please make sure that you know the menstrual cycle. They can always ask it at any time. These are the flow chart of the different hormones, and that you can just go through. It's everything that I basically just explained. But what I want to mention to you is that remember FOLP, and then also you need to know the negative feedback of progesterone and FSH. Okay, so that is basically the pathway that is saying that when there are large amounts of progesterone, when progesterone is high, it inhibits the secretion of FSH by the pituitary gland. When progesterone is very high. Now, let me ask you, why would progesterone be high? Or when is progesterone high? When pregnancy has occurred. Why? Because the progesterone needs to maintain the endometrium for implantation or prepare the body for the pregnancy, right? So then progesterone levels will be high. So when progesterone levels are high, why does it inhibit FSH secretion? Because FSH is going to stimulate follicles to develop, and if there's already fertilization, we do not need that to happen. Right, let's quickly do a question. Basal body temperature is the body temperature while a person is at rest. Okay, so your basal is like your resting body temperature. An increase above the basal temperature occurs immediately after the day of ovulation. The basal body temperature method can be used by women who want to fall pregnant. The graph below represents the body temperature chart of a woman with a 28-day cycle. So they are saying immediately after ovulation. Okay, so when is ovulation? At day 14. This is what you need to talk to yourself about when you read passages. It's saying that the basal temperature is at rest. But now, after ovulation has occurred, they notice that the basal temperature has increased. So now you can think to yourself, why would this be used for women that want to fall pregnant? Ask yourself that. Because if their basal temperature then increases, it implies that ovulation may occur. Right. So if ovulation may occur, then that is a very good thing for them to realize that, okay, my basal temperature is high now, which means that ovulation has occurred, which is a greater chance for me to fall pregnant. Why? Because if ovulation occurred, there will be an ovum. And if there's an ovum in the fallopian tube, it can be fertilized. Right. So let's look at this chart. Okay, I've got the chart on the next page as, oh no, I don't, with the questions. I thought it was here. So let's have a look, 28-day cycle. Here we've got day 14. Okay, so that's at day 14. You can see here that the basal temperature, they can see that this is the basal one. It shows you here where's the basal temperature. Okay, so you can see over here that it's fluctuating up and down around till day 14. And then after day 14, it's still there, around about this day here. It starts to go up. Right. So that's just how you need to start analyzing your diagrams, your graph, sorry. Right. So what is the basal body temperature? We just looked at it now. It's 36.2 degrees Celsius. The day of ovulation. When is the day of ovulation? Now you know that it's 14, but they're asking you according to the graph. Remember, it's around day 14. So you cannot say day 14, grade 12. So you have to go and look at it. Sorry. Let's go back there quickly. Right, you need to go back there. So you need to go and look there. Okay, no, day 14 was still, at day 16 it started to increase after day 16. So that means day 16 was the day of ovulation. Using evidence in the graph, give a reason for your answer. Okay, so it's exactly how I explained to you from the graph. You look at the graph and you say that the body temperature increased above the basal temperature immediately after day 60. Okay. Progesterone is the hormone that causes an increase in the basal body temperature. Now, I quickly want to stop here. They are giving you information here. They are telling you progesterone is the hormone that causes an increase in the basal body temperature. It is the progesterone that does that. Now remember, they said it's after ovulation. So the progesterone does that. Now I want you to ask yourself, where does that progesterone come from? Okay, but that's not the question. The question wants to know. Oh, sorry, there's a mistake here. This is actually the question. Okay. Explain why the increase in basal body temperature occurs only after ovulation. Okay, so why? It's because progesterone is only available after ovulation. Why? Because it gets secreted by the corpus luteum. Right, so now you've got to say that the corpus luteum is formed and it secretes after ovulation, which secretes progesterone. So that is why there's an increase in the basal body temperature only after ovulation. Okay, next question. Explain why the basal body temperature is measured when a person is at rest. So why are they saying you're measuring it at rest? And the reason why is because physical activity is going to cause the body temperature to change. Okay, you know, once you've been exercising or you do some strenuous activity, that's going to affect your body temperature. So it has to be at rest when your body is calm and you're not really going through a lot of processes, and that's when you can measure what the basal is. Or you can say the other side of the coin, where if you have a low body temperature, it can change your body temperature as well. If you have a low physical activity is when you have, you can also have preventing a change in the body temperature. It's not going to give you a true reflection. What I want to show you here, grade 12, is that explain means a cause and effect. Okay, so here simply the corpus luteum is like, you can say it's the cause. What is the effect? It secretes progesterone. In this one, also explain cause and effect. The physical activity changes the body temperature. The cause is the physical activity. The change is the change in the body temperature. Right, then quickly going to the development of the zygote. Very important. You need to know fertilization. You need to know what fertilization is. So please go through all of these. You should know this, how fertilization occurs. And you need to know that it's the head of the sperm that is going to penetrate, okay? Because the acrosomes at the head of the sperm cell, they are going to dissolve the membrane, and then it's going to, the head will enter, and the neck part and the tail will not enter. Okay, and then the membrane becomes impenetrable to other sperm cells. Why? Because one has already entered and was successful. So we do not, it prevents there from being multiple sperm cells entering, right? And then we have the nuclei of the ovum and the nuclei of the sperm cell are going to fuse, and we're going to form a diploid zygote, okay? Then the zygote is going to go through all of their different processes. They are going to undergo mitosis to form 4 cells, 8 cells, until it forms a ball of cells called a morula. It's going to go through further mitosis until it becomes a hollow ball of cells, which is called a blastocyst. And that blastocyst is going to move along the fallopian tube until it reaches or implants in the endometrium. Why I'm going through this is because grade 12, many learners don't understand that it's mitosis that is taking place along all of these stages. Please know, you also need to know from zygote, it becomes a morula, becomes a blastocyst, okay? Right, we have another, it tells you here another question. It says this is the human embryonic development. So we're having a look at the diagram. Let's see what's going on here. We've got a sperm cell and we've got the structure. And they're showing us here that these two come together, which means that this must be the ovum, right? So when the sperm cell and the ovum are going to, the nuclei of these are going to fuse, what's going to happen? We're going to form a zygote. Then we're going to have a morula. Then we're going to have a blastocyst, and then we have the embryo. Okay, so you need to also understand the diagrams. So cell B there, as I said, we just said now it is the zygote. Even though it looks, I want to show you something. B and A look the same, right? So the reason why you need to analyze the diagram is because of that very fact that A and B look the same, but it's because of the positioning of the different structures and the arrows. That is what's going to guide you to which one is the zygote and which one is the ovum. Structure C is the blastocyst. How do we know? There's a hollow over here, a hollow ball of cells. What's the difference between A and B? Okay, so you see now they're showing you. You can look at them and you see they look exactly the same. But now you know, the one is the ovum and the one is the zygote. What is the difference? Well, definitely A is haploid and B is diploid, okay? And cell A is not fertilized, whereas cell B is fertilized. What's the fluid surrounding the embryo? Okay, so remember the embryo is going to develop, the fluid surrounding it is called the amniotic fluid, yes. And what's the 3 functions of the amniotic? Name 3 functions. There are more than 3, there are 4. But they ask for 3. So we'll say allows for free movement, protects the fetus against mechanical injury, prevents dehydration, and prevents temperature changes. I want to take something quickly about this before we move on. I didn't mention it before. Exam techniques, grade 12, very important. 3 functions. If they ask you for 3, you give them 3. The marker is only going to mark the first 3. That is a rule of marking, of a principle of marking. Okay, so if they ask you for 3, they will mark the first 3, correct? The first 3, and they'll disregard the 4th one, even if the 4th one is correct. Okay, if they just ask you, state all the functions or state the functions, then you can list 10 of them, and the marker has to read all of them and see which ones are correct. It's very important that you read the instruction and you answer the questions that are asked of you. Right? Then you need to know that after implantation, what happens? You need to know how this embryo is going to develop. You need to know all the extra embryonic membranes. You need to know how the placenta forms. I'm quickly going to show you that one. It's another place where learners get wrong. So what happens is that the chorionic villi that is going to develop from the chorion, they are going to be embedded into in the endometrium. So that endometrium plus the chorionic villi is going to form the placenta, and this is attached to the uterine wall. Very important. You also need to know the functions of the placenta. So this is a summary of a fetus in the uterus. All the structures are there. Please make sure you know the structures, you know the functions. This is a very lovely slide which can show you all the structure and the function of all the different parts. Make sure that you know them. Make sure that you study them. And make sure that you know how to label this structure. Okay, so that is all for those two topics. Now we're going to have a quick 5-minute break, and then my colleague Taniko will take over for the rest of the lesson. Thank you.
back, grade 12 learners, and special thank you to Vedika, my colleague, for unpacking so much of paper one content. We have a lot of topics in paper one, but she's covered a lot of them graciously so, and for that we are grateful. Now let us just carry on. We are going to look at the endocrine system. And the endocrine system with homeostasis together, they make up 34 marks. So it's a lot. It's a lot of marks. So you better know your story well so that you get your marks. If we look at the endocrine system, basic things that you need to know, especially with this diagram, it is giving you an overview of what needs to be known in endocrine system. I would advise you as you do your revision to know the position of the gland and to know the name of the gland, to know the name of the hormone or hormones that are secreted, and to know the functions of each hormone. This is going to give you a clear, well outlined, annotated diagram. And this diagram is going to help you to holistically study endocrine system. Anyway, we are not learning, but we are quickly looking at what must be remembered for examination purposes. So we have the pituitary gland. We have our pituitary gland, which is, and please don't quote and call it master gland, but pituitary gland is a master gland because it controls many hormones that are secreted by it, which control other parts of the body. But another name for the pituitary gland, some of you might know the hypophysis. Right? So pituitary gland or hypophysis. The hypophysis is accepted in the examination. I want you to just quickly revise that there are quite a number of hormones. We've got the growth hormone, which is abbreviated as GH. We have got TSH. Please be careful. It is TSH and not STH or otherwise. Learners have a tendency of getting confused in the exam, and they can say anything which does not mean thyroid stimulating hormone. And we've got FSH. Again, it's FSH, not SFH, because if you say SFH, then you are meaning something else. That is follicle stimulating hormone. We have LH, and we've got prolactin. Know the functions of those hormones for your examination. We also have, and you know the position, so you must know where the pituitary gland is specifically found. We have the thyroid gland. You can see where the thyroid gland is located, which secretes thyroxine hormone. Hey, grade 12 learners, do you remember the negative feedback mechanism that exists between the TSH, the thyroid stimulating hormone, and the thyroxine? You must know what happens and what brings about the response of the pituitary gland to secrete less or more TSH. And you must also indicate the link on the thyroid gland being able to secrete more or less thyroxine. This is your marks. Understand this so that you get your marks. We have got the hypothalamus, and remember the hypothalamus secretes ADH, and ADH has everything to do with our osmoregulation, the control of or the regulation of the amount of water in the body. We have the adrenal gland. Please check the position. There is the adrenal glands that are responsible, and they are found at the posterior end of the kidneys, right? And they secrete two hormones, and the two hormones are, do you remember? It's adrenaline. Adrenal gland secretes adrenaline hormone. It's a gland adrenal, and adrenaline is a hormone. And this one is responsible for the flight or fight mode, emergency state. And aldosterone hormone, aldosterone, that has to do with the regulation of the salts in the body. We have the ovary, which is found in females only, which secretes two hormones, which are called ovarian hormones. So in the ovary, we have got structures which Vedika spoke about, the structures that will secrete estrogen, which is a, a hormone, and also progesterone, which is a hormone. I hope you remember that estrogen is secreted by the developing Graafian follicle, and where is it found? Inside the ovary. And the progesterone is secreted by the corpus luteum. And again, this is a hormone that has the two hormones have their specific functions in reproduction. Thanks, Vedika, that you have clearly outlined this information for us. Then with male, by the testes, we have got testosterone. Please remember to know the correct spelling. Testosterone. Also practice with how your cheeks and your lips are going as you do, or as you say, testosterone, for the correct spelling, because learners always ask, does spelling count? And yes, this is one of the examination tips. Spelling counts a lot. Question 1.2 in your question paper, that is all about terminology, has everything to do with terminology, meaning that the spellings must be correctly outlined as you answer your work. I reserved this one, the pancreas, as the last one, because I do have special interest on how to revise this. The pancreas, please just know that how it is, and remember that the pancreas, it's the only gland that is declared as the exocrine and also endocrine. So this is an exocrine and endocrine gland. And why are we saying this? Because the pancreas, remember, has got ducts. So when it comes to digestion, the ducts are carrying the pancreatic juice, which is secreted by the pancreas, to the intestine to complete the process of digestion. However, the pancreas is also the endocrine gland because it secretes two hormones which work opposite each other, and the hormones are glucagon and insulin. Glucagon and insulin. Yes, learners, remember the confusion that you always have of not knowing the difference between glucagon and glycogen. Glucagon is the name of the hormone. Glycogen is the state in which the excess glucose is stored in the liver cells and the muscle. Let us look at some questions. This is from June 2025, June, a paper. Right, so the question says, read the extract below. Insulin pumps in people with diabetes mellitus. I wanted to mark diabetes mellitus. It's a sugar diabetes. The beta cells in the pancreas are unable to secrete sufficient amounts of insulin. These people are treated with regular injections of insulin. Electronic pumps, so you are reading and you are marking electronic pumps, have been developed as an alternative to injections. They pump insulin into the blood according to the different blood glucose concentrations. The pump can be clipped to a piece of clothing or fastened to the skin with an adhesive patch. These pumps then infuse insulin through the skin into the blood. Wow, technology is working for us. We come a long way with insulin making. Remember way back in the past, before technology came into picture, insulin was extracted from pigs and cattle, meaning that there was a lot of killing that was taking place, and we moved now to insulin injection. So people that suffer from sugar diabetes, they inject themselves. Look at how advanced we are coming. Now that we are talking about the electronic pump, as Vedika indicated, when you find extract, you don't read the extract once. You read and you read again and read again so that you understand. Underlining those things that stand out for you, and they will assist you to answer questions. Another advice: when you find an extract, do not answer questions in isolation. What I mean is that look at the first question, look at the second question, look at all the questions that are packaged for that extract, so that you don't contradict yourself, because some answers we give before we get to the actual question. The first question is: identify a group of cells in the pancreas that contain beta cells. So we are looking at a group of cells in the pancreas that contain beta cells. For your information, we have got beta cells and alpha cells. However, it is not very crucial for you to really remember the individual names, but we must know this group of cells in the pancreas. And what is our answer? Everybody should be knowing that long one. It is called the islets of Langerhans. Islets of Langerhans. So basically, the islets of Langerhans, they have got beta cells, and they also have got alpha cells. So they've got alpha cells. Right, I still repeat, for you to know the names, it's not crucial. That is why on the extract, beta cells are mentioned for you. The next question is: identify. It's still on the extract. Identify the question B, the signal for the pump to turn on and start pumping insulin. What is it that influence the pump to start pumping insulin, which will be infused to the bloodstream of a person? And what are the indicators? The signal is a high concentration of glucose in the blood. A high concentration of glucose in the blood. Remember the functions of insulin and glucagon. Insulin, its responsibility is when the blood glucose is above normal. The blood glucose, not just glucose, but the blood glucose. So we are talking about the blood sugar. The blood glucose, when it's above normal in the blood, so it's in excess, that is when now insulin, under normal circumstances, when we do not have the need to substitute the natural functioning, the insulin is going to be secreted. By the way, what is the name of the gland that secretes insulin? It is the pancreas, and the special cells are the islets of Langerhans. So the insulin is secreted under normal circumstances, so that insulin can do what? It will convert excess, what is not needed by the body, excess glucose, now into glycogen, which can be stored. So here, the signal for the pump to be turned on, remember, it's now clipped on the hand. What is it that's going to inform the need for the pump to be turned on? It's now when we've got a high concentration of glucose in the blood. I hope the explanation is making a lot of sense. The next question is: state why the cells identified in question 321 have endocrine characteristics. Which cells are we talking about? We are talking about the islets of Langerhans. Why are they having the endocrine characteristics? The good news is that even before we came to the question, we have quickly revised and reminded each other or one another about what endocrine system is and also what the exocrine system is. So we know by now why we are saying that the islets of Langerhans have the endocrine characteristics. And let us look at the response. It's number one, because the endocrine characteristics or endocrine system secrete hormones. So the islets of Langerhans, they secrete hormones. In this particular case, for you to mention the names of the hormones is fine. But if we're not given the extract, which is giving us a specific scenario, you don't have to mention the names of the hormones. But because we are confined into the extract that was given, we are free to mention in our answer that insulin and glucagon. So we've got insulin and glucagon. These are the hormones that are secreted. And another indication, the reason is that the hormones, remember, what is the transport system for the hormones? It is the blood system. So it's secreted and it's directly into the blood. So it has no duct, or it is ductless. The next question is, describe the effect of insulin on the liver and the muscles. Remember the relationship that we have of the liver and the muscles, and the stored excess glucose in the form of glycogen. So you need to understand this full relationship that exists. And now you are describing for us, because insulin stimulates the conversion of glucose and glycogen. So in other words, if we've got too much of glucose, the insulin is going to be now influencing the conversion of glucose into glycogen and stored. But now, if there's a need in the body, what will happen in the body is that now glucagon as a hormone is now going to stimulate. I want to clarify something. Insulin does not convert. Insulin does not convert glucose into glycogen, and glucagon does not convert glycogen into glucose. They stimulate the conversions thereof. Please be careful of that. And also, we are talking about the effect of insulin on the muscles. The insulin promotes the absorption or the usage of glucose by the cells. Remember that when glucose is found in the blood, it must also be absorbed into the cells so that a person has got full functionality. So insulin is there to do what? To influence or promote the absorption thereof of glucose into or to the cells or by the cells. The next question is, explain why electronic pumps are more effective in controlling blood glucose levels than insulin injections. Already, me and you, let's reason together. If you are using an injection, you are given a specific time that you are going to inject yourself. So whether the glucose, the blood glucose is high or low, you inject yourself because that's a norm. But now with this advanced technology, what is it? Why are we saying now the electronic pump is more effective? And then let us see why it is more effective. Because it delivers insulin specifically according to the glucose level. So in other words, it's only when there's a need for glucose to be, for blood glucose to be pumped in the blood, that this kind of the pump will be activated. And also to allow for better blood glucose management or regular dosing. I think I have outlined when I said that with injections, regular dosing is compromised because you are given a certain amount that must be injected, whether there's a need at that particular time or not. Let us quickly look at homeostasis, which is about the skin. The question is, describe the role of human skin in regulation of body temperature. So we're talking about the body temperature, but more specifically on a cold day. With homeostasis, which is based on thermoregulation, we will look at a hot day or a cold day. However, examiners are migrating into more advanced approach of asking questions. So sometimes they will not say it as is, that it's a cold day, but they can give you a scenario indicators that will make you deduce from them that this is a situation on a cold day. Let us see what will happen. Now, on a cold day, the cold receptors in the skin, So we're talking about receptors. Once more, Mrs. Maharaj spoke about receptors when she was explaining the response to the human response to the environment. So we're talking about receptors. Where are they found? In the skin. What they do, they convert the stimulus into an impulse. Key to us, you must know that we're talking about the stimulus, which is now converted into an impulse. Now, what will happen? This impulse is then sent to the hypothalamus. I want you to remember that it's not the hypothalamus that converts the stimulus into an impulse, but the cold receptors, and you must specify where they are found. Then this is going to stimulate vasoconstriction. We know vasoconstriction, it means that the blood vessels of the skin. Don't just say blood vessels. We've got blood vessels all over our bodies. You must specify of the skin, constrict, and then let blood flows to the skin and let blood flows to the sweat glands. Therefore, less heat is lost. You see, I'm underlining less because less is, you must tell us it's less, less, less. Less heat is lost from the skin through the process of radiation. And now the skin, the sweat glands are stimulated to produce less sweat, and less heat is lost through evaporation. Please remember the indicators. There's no point for you in memorizing homeostasis if you don't know. In a cold day, it's less, less, less for less. And it's more, more, more for cooling down of the body. So the opposite will happen if it was on a hot day. Please be careful because learners lose marks unnecessarily on this question. Let us look at plant response to the environment. And this question has 13 marks. It's a lot as well. So let us look at some, it's going to be one example of a plant response to the environment. But for your information, remember the terminology around plant response. If you need to know tropism, you must know phototropism. You need to know geotropism, right? And the names of the different hormones, gibberellins, abscisic acid, and auxins, which form part of the plant hormones, right? So an investigation was conducted to determine the response of plant seedlings to light. The procedure was as follows. Grain seeds were allowed to germinate into seedlings. So there's germination that is allowed. One half of the seedlings were planted in one small pot and the other half in another small pot. So how many pots are involved here? 2. When the stem reached a height of 1 centimeter, the pots were placed on the discs of 2 clinosets. Right, a clinostat, now you are being told what it is. A clinostat is an apparatus consisting of a slow rotating disc when switched on. Both sets of apparatus, A and B, were placed inside boxes with openings on one side only for light to enter. By the way, the light that enters from one direction, we call it unilateral light. One clinostat was switched on, the other one was switched off. This is key for you to remember. And the apparatus was left for how long? For 2 weeks. Read again for you to understand. I'm not reading with you, but you read again. Now we are looking at the setup of our apparatus. You can see they are showing us the clinostat disc. So this is the clinostat disc. You can see that we have the same one here, right? So we've got apparatus A and apparatus B. It is very important for you to be aware of your surrounding in terms of what you are reading. You can see we've got 2 boxes. This is a box and this is also another box. You will also notice that this is now the opening that was coming from one side only. And these are our seedlings that were left for 2 weeks to grow. You also check how they look. First question is, identify the plant growth response demonstrated by the result. So already at the back of your mind, you know we are talking about light because it is indicated unilateral light. So what is the response there? It's phototropism, the response of the plant growth towards light. In which apparatus, A or B, was the clinostat switched on and the disc rotating slowly? So what you do, you come to the apparatus and you check. You need to be able to have a good background in order to tell. So which one between A and B was the clinostat switched on? And our answer is B. So you look at B. So this one, this apparatus B here, so the whole set here, the clinostat was switched on. The good thing about life sciences, you explain the answer for your question. So give us explain the answer, and the explanation is that all the seedlings grew straight up or showed no phototropism because they were evenly exposed to light. Look at evenly exposed to light or not exposed to unilateral light. Or there is also equal distribution of auxins. You can see that the seedlings here, they've all grown up straight to show that there was no to show that there was a movement of the clinostate. So they were not only exposed to unilateral light. Beautiful. And in that process, you can already tell what is happening in apparatus A. But let's look at this question. Explain the growth response of the seedlings in apparatus A. So now we are looking at A. You are going to explain it. Always remember to check the verb that is used for the question and also check the mark allocation. This is a lot. 5 marks is up for grabs and you can score them with ease. And let's have a look. Because the seedlings or stems grew or bent to the left. So they can see they are bending to the left, right, towards the light, because the light is entering from this direction into, but it's unilateral light. Because they were exposed to light from one side or unilateral light, and auxins diffuse to the right. So you know that auxins will diffuse to this side or to the dark side of the seedlings. The high concentration, check the high concentration of auxins on the right or on the dark side stimulates cell elongation or cell division or growth on that side. So the logic is that once we have more auxins on the right, there is stimulation of cell elongation. Hence, you can see the seedlings are bending towards the light. I hope you understand when we speak about geotropism, because that is about the response of the plant based on gravity. Let us look at reproductive strategies. With reproductive strategies, this has 8 marks in the examination. The chunk or the most marks are coming from human reproduction, but you also need to know this. For this, just a quick reminder that we've got ovipary and we've got ovovivipary and we have got vivipary. These are the reproductive strategies. Just remember as well, ovipary, eggs are laid and the development of the embryo is outside. Ovovivipary, the eggs are not laid, but the development of the embryo happens in the inside of the female, and the birthing process takes place inside, so the hatching takes place inside, and then the young ones are born alive. Vivipary, an example of vivipary, development is inside the female, and the nourishment is from the placenta. You have just done this in human reproduction. Check the type of egg that is produced, check the type of nutrition that is needed, and also see if where the development of the embryo is taking place to avoid confusion. Then we've got the 2 easy comparisons: precocial development, altricial development. And maybe for your information, remember precocial development takes place when the young one is born, is able to fend for themselves, they can walk around. Hence, we say that the body is well developed for precocial and the eyes are open and there is fur and maybe feathers, right? And please avoid saying hair. This fur or feathers. And there's a low degree of parental care required because when they are born, they are able to run around. Young can move soon after birth. And altricial development, it's exactly the opposite of precocial development. The young ones are underdeveloped, their eyes are closed, and they are usually naked. A high degree of parental care is required, and the young have limited ability to move freely. Just imagine your baby sister at home or your baby brother or your cousin. When they are born, they can't do anything. They are banking on the full parental support, from changing of the diaper to feeding them and to checking their temperature when they are not feeling well. And on that note, grade 12s, we just want to say that thank you so much. And for your information, please tune in tomorrow, the 19th of September, 2:30 4 o'clock for paper 2 revision. Proper planning always prevents poor performance. And we are saying to you with love, and we hope that you are going to nail it as you sit for your examination tomorrow. Thank you so much.